So the next unit that we are doing is talking about cellular reproduction. I will let you guys know that I'm actually breaking this section. It's all one set of notes, but I am breaking the videos into three just so that it's not quite so overwhelming. So this is the first of the three. So we have two different types of cell division which result in reproduction. So we have asexual reproduction, in which a parent divides into two genetically identical daughter cells. It's essentially making a clone of yourself. This is how most of the bacterial world works. And we have sexual reproduction, in which case I have two parents contributing genetic material to the offspring, and that offspring is genetically unique from either parent, and also, with the exception of twins, genetically unique from any siblings. Now, cell division has three main roles, okay? I replace lost or damaged cells, okay? When you cut your skin, your body heals. That's what this sort of falls under. I have growth. Multicellular organisms grow and develop from a single cell, okay? That's really one of the amazing things of life. We all started as a single cell, and then we become literally tens of trillion cells. And then cell reproduction is the third role of cell division. Okay, so our example here, cell replacement in our skin, right? Your top couple layers of skin are actually dead and they're constantly being replenished by the cells beneath it. Prokaryotes, so pro means before. So prokaryotes are not us. Prokaryotes are typically bacterial. Basically, in this case, I have genes that are carried on a single circular piece of DNA. DNA has few proteins and is attached to the plasma membrane. It is not membrane bound, okay? There is no nuclear envelope in prokaryotes, and the cells divide by binary fission. Now, the lack of a nuclear membrane is actually really important. Bacteria honestly wants to mutate because that's the only way it's going to become different, because it reproduces asexually. Okay, so in order for that to happen, we honestly want less protection of our genetic material. So in binary fission, an exact copy of the chromosome is made. Okay, the attachment point divides, so the two chromosomes are attached at separate parts of the membrane. Cell elongates. Okay, I've got additional plasma membrane that is added. And then in the end, I have division. And you'll know here in binary fission, we end up with two daughter cells that honestly are the same size. So growth occurs before division is complete. This is different than budding, which is budding, not your rear end. Anyway, in that case, a little bit, the DNA basically just encapsulates in a small portion of plasma membrane, and then it grows after division. Binary fission, the growth occurs before division. Eukaryotes are us, okay? So we have our genome. Our genome is our total hereditary endowment of DNA, okay? It's everything. It's all 3.5 billion base pairs, which we have in every single one of our cells because we started as a single cell. So before jobs can take place, you need to make sure you have all the genetic information available for that. Genome is species specific. Okay, there are variations in our genomes. They can be very small amounts that are different between species. And in fact, it really is shocking sometimes when we look at species and see how many similarities there are in between species. But each genome is actually species specific. Human DNA, if we were to uncoil it all, actually extends about 3 meters. So, close to 10 feet. So you're talking about a lot of information to get evenly distributed. We package DNA into chromosomes, okay? They're simply more manageable that way. And we'll talk about this in a moment. So eukaryotic chromosomes, it's made of chromatin, okay, which is a combination of DNA and protein molecules. If you are looking at a cell in interphase, okay, which is the in-between part, they're not dividing yet, you actually cannot see the DNA. Okay, it only gets packaged up when it comes time to divide. You'll notice here these histones, they're sort of like, I like to think of them like the spool on the inside of a strand of thread. Okay, it sort of allows the DNA to be able to wrap around it. So before the cell starts dividing, the chromosomes are duplicated. Okay, we make copies of it before we package it up. It just is easier that way. And this process produces sister chromatids. Now, we've got chromatids and chromosomes and a chromosome. People get awfully confused. So, sister chromatids. This is the exact same copy, okay? So I inherited one chromosome from mom and one chromosome from dad. Sister chromatids is my chromosome from mom has made a copy of itself. My chromosome from dad has made a copy of itself. Okay, when I'm talking tid, I'm talking identical. Okay, so when the cell divides, the sister chromatids separate. Two daughter cells are produced and each has an identical set of chromosomes. When I cut myself and my skin cells divide in order to fill in that cut, they're not genetically different from each other. They are genetically identical to one another. Human somatic cells, that means our body cells, contain 46 chromosomes found in 23 pairs. You should know this because I beat it into your brain in 6th grade, and Lord only knows that Mr. Jordan probably went ballistic on you in 8th grade with it. Plus, you know, Mrs. Tompkins, all that stuff. Our gametes have 23 chromosomes, okay? Half the number. It's very important so you don't get Blinky the three-eyed kid. So mitotic cell division. So first of all, we've got mitosis and meiosis. Mitosis happens in mito. Okay, silly as that is how I remembered it since, you know, 9th grade biology. In a dividing cell, I have interphase, which is just chilling like a villain, and then I have growth phases. The mitotic phase is usually the shortest part of the cell cycle. Interphase accounts for about 90% of the time the cell exists. Most of the time, it's just hanging out. It's not actually dividing. There are subphases of interphase. So I've got G1, and this is the cell grows, and I produce proteins and cytoplasmic organelles. Cytoplasmic organelles are things like ribosomes, Golgi apparatus, mitochondria, stuff like that. The S phase stands for synthesis. I can talk, synthesis. So this is the duplication of chromosomes. I'm synthesizing my DNA. I'm making a copy of it. And then G2 is simply a longer growth phase, okay, while I get ready for cell division. Because in mitosis in eukaryotic cells, just like in binary fission, my cell basically doubles in size before I cut it in half. So in G2 of interphase, I can see my nucleus. I've got a nuclear envelope there. Now, I might contain one or more nuclei. Okay, so I might actually, now my nuclei have formed and separated from one another. I've got centromeres. Okay, centromeres are here. Fun fact, we have no idea what they do. They only show up in animal cells. If we take them out, cell division still occurs. So we really have no idea what the role of the centromere is. And the chromosomes can be seen. Okay, I'm sorry, the chromosomes are duplicated. Basically, I cannot see the chromosomes at this point. It's still just a googly mess in there. Mitosis has 4 phases, pro, meta, and telo. Okay, so prophase, this is when I begin to be able to see my chromosomes. Okay, my chromatin fibers become tightly coiled. My nuclei disappear. Translation, my nuclear envelope disappears. Okay, chromosomes appear as 2 identical sister chromatids joined together by a centromere. Okay, the centromere is simply the center part of a chromosome. And the centromeres move to opposite poles. Okay, or I'm sorry, the centrosomes move to opposite poles, not the centromeres. By the way, these pictures up here are created through something known as FISH, which is fluorescent in situ hybridization. You have different molecules that glow, like what's found in, say, fireflies or jellyfish or the anglerfish from Finding Nemo. Anyway, so the blue here is my DNA. This green, they're actually spindle fibers, which we're going to talk about in a second. And the red that you're seeing is actually the cytoskeleton. So my nuclear envelope disintegrates. The microtubules of spindle extend from the poles, which are opposite ends of the cells, and then they attach at the centromeres. Okay, these are my spindle fibers. And I have a kinetochore that forms. It's this very, very defined, basically where the centromere was. It's this very defined region, and the spindle fiber actually attaches to the kinetochore. Now, metaphase. Metaphase, in my opinion, is the easiest one to see because they all line up in the center. Honestly, and I'll show you later, it looks sort of like a black Sharpie marker when you look at it in a stained cell. So centrosomes are now at opposite poles. The chromosomes convene at the metaphase plate. Okay, when they're aligned, sister chromatids straddle the metaphase plate, and I've had completion of my spindle fiber formation. And again, you can see it here. You can see all these blue are now lining up in the center. In anaphase, my paired centromeres begin to separate. Each chromatid is now considered a chromosome and moves to the opposite pole. So these two are exact copies of each other. These two are exact copies of each other, and they're moving to the opposite pole. You'll note here, guys, I just like to say, like, these two are probably the same chromosome. Say this one came from mom and this one came from dad because we're paired. Whereas these two are also, again, probably the same chromosome number, but these are exact copies. You know, say, for example, this chromosome here, this is an exact copy. These are exact copies, but these are not exact copies. And we'll talk about homologous chromosomes versus chromatids and stuff later. So chromosomes move to the poles in anaphase, and my microtubules shorten. So basically, I've got this little motor protein, and it chews up and spits out the microtubule, which is the spindle fiber. And these subunits can be used again. It's sort of fun. Telophase and cytokinesis. I have microtubules that actually sort of stretch the cell out. They make this, so these are not ones that are associated with my separating chromosomes, but they sort of stretch it out. The daughter nuclei form at two poles of the cell. Okay, so now I'm getting the reformation of my nuclear envelope. And chromatin fibers begin to become less tightly coiled. And cytokinesis is actually the division of the cytoplasm. So in animal cells, I get a cleavage furrow, which is basically it just pinches off. Okay. It typically occurs during telophase, and it is the division of the cytoplasm. It is different in plant and animal cells. Okay, in animal cells, I get a cleavage furrow. In plant cells, my cell plate forms. And basically, this is part of the cell wall. The cell plate goes and it extends here. And then once it cuts this in half, the membrane will simply spontaneously form on the inside. So during telophase, like I said, this is where my cell plate forms. Okay, and I sort of went over that in the last picture. And this is just looking at it from a microscope perspective. This is interphase. I can't see anything, right? There's nothing exciting there. When I start seeing little squiggly wormy things, that's prophase. Metaphase, told you, it looks like you drew a thick Sharpie marker. This is anaphase, telophase, and then cytokinesis. I would like to point out, folks, I am never, ever, ever on a test going to ask you to differentiate between anaphase and telophase. It's hard unless you're going to get into cell biology, which this is not. Okay, I'm just letting you know. I'm not going to make you go, oh, wow, I think that this is, if they're over here and it's a little bit closer, is that anaphase or telophase? I'm just not going to do that to you guys.